So our protocol allows us to prepare enantiopure epoxy fatty acids, and this method might help advance research on the mechanisms by which these metabolites act in the body. Historically, pure epoxy fatty acids are very difficult to obtain, so our protocol is simple, rapid, cost-effective, and it allows us to prepare significant quantities of these metabolites.
Yes, we developed a protocol for DHA, but it also works very well for EPA and arachidonic acid. Work quickly, as some of the reagents are air sensitive, specifically DHA and NADPH. And be sure to prepare everything in advance so that setup is simple. Also, be aware of the hazards. Our protocol uses techniques in both enzymology and organic chemistry. Visualization is critical to aid readers and viewers who aren't familiar with the specialized techniques in these fields.
To inoculate, use a sterile loop or pipette dipped in glycerol stock of PBS-BM3 transfected DH5-alpha E. coli into 5 milliliters of sterile LB broth supplemented with 0.5 milligrams of ampicillin. Incubate the cell culture in a shaker at 37 degrees Celsius for 24 hours at 200 rpm. Then, add the 5 milliliter starter culture and 100 milligrams of ampicillin to 1 liter of sterile LB broth in an Erlenmeyer flask. Shake at 37 degrees Celsius for 6 hours at 200 rpm, then at 30 degrees Celsius for 18 hours at 200 rpm. After that, collect the cell culture into centrifuge tubes and centrifuge the cell culture at 4 degrees Celsius for 10 minutes at 1000 times G. Pour off the supernatant into another flask. Decontaminate with bleach or using the autoclave.